Assalamu alaikum everyone. Myself Pardeep Singh Rajput from final year Botany Department, University of Sindh. Roll number 2K17, Botany 137. I am thankful to Sir Professor Dr. Abdul Aziz Ujan to give me opportunity to present here on phytochrome mechanism in plants. Today's my topic is on phytochrome mechanism in plant. First of all, we discuss about introduction. Phytochrome is a photoreceptor, a pigment that plant and some algae and fungi use to detect light. It is sensitive to light in the red and far red region for the visible spectrum. Biochemically, phytochrome is a protein with a billion chromophore, the pigment that control growth and flowering in many plants. History and discovery. The first important work in the field was conducted in 1930s by L. H. Flint and E. D. M. S. Elster. In 1935, they reported that the seeds of Lactuca sativa did not germinate in dark but showed very good germination when exposed to wavelengths from 525 to 700 nanometer in the red regions. H. A. Broadwick and S. B. Hendrick and other 1952 of the U.S. Department of Agriculture Plant Industries stations in Beltsville, Maryland, using large spectrograph confirmed finding of Flint and M.S. Elster. Now we will discuss about long day, short night. A diagram of long day, short night, short day, long night, short day, interrupted night, short day, red interruption, short day, red followed by far red. ये जो डायग्राम आप देख रहे हैं ये उन प्लांट्स का है जो लॉन्ग डे प्लांट होते हैं या शॉर्ट डे प्लांट होते हैं और शॉर्ट नाइट प्लांट होते हैं उनको मद्देनज़र रखते हुए ये डायग्राम शो हुआ है। तो आपको डायग्राम में दिखाया जा रहा है उनके बारे में। तो शॉर्ट डे प्लांट्स क्या होता है? शॉर्ट डे प्लांट्स फ्लावर ओनली व्हेन नाइट्स आर sufficiently long, when long nights are interrupted by a short day of white light. Flowering is again delayed. The active wavelength for the light response was found to be red light. Moreover, the effect of the red light treatment could be suppressed by treatment with far red light. Suggests the existence of receptor protein that is activated by red light and inhibited by far red light. Now we will discuss about the distribution of phytochrome. Presence of phytochrome has been known as angiosperm, gymnosperm, liverworts, muscle fern, and some green algae. Distribution of phytochrome is grass-short is variable, but oat, ray, and barley seeding all have high concentration in apical regions of the poliopetal near the shoot apex and in the growing leaf bases. Phytochrome is present in most organic of the all plant investigation, including roots. In green lower plants, phytochrome is permeability localized in conformation in or near the plasma membrane. Characteristics of phytochrome. The action spectrum of the light needed for these responses shows a peak in the red at about 660 nanometers. These responses can be reversed by an application of far red light soon after the red treatment. Sensitive spectrometer can measure a decrease in absorption at 730 nanometer when sensitive plant tissue are exposed by red light. The change in absorption is caused by conservation of photoreceptor from one structural form to another structural form. The red absorbing form changes to the far red absorbing form when it absorbs red light and back again when it absorbs far red light. Phytochrome has two different chemical structures that are interconvertible. The form are named by color of light that they absorb maximally. Number 1, the PR form. The PR form absorbs at the peak of 660 nanometer. It is the form synthesized in dark growing CD. When PR absorbs red light, it converted to the PFR form. जब PR एब्जॉर्ब करती है रेड लाइट तो वो कन्वर्ट हो जाती है PFR फॉर्म में। आगे चलते हैं तो नंबर 2, द PFR फॉर्म एब्जॉर्ब एट द पीक ऑफ 730 नैनोमीटर। मीन्स जो PR फॉर्म होता है वो 660 नैनोमीटर पर एब्जॉर्ब होता है, जबकि PFR फॉर्म है 730 नैनोमीटर पे जो है एब्जॉर्ब होता है। द PFR इज ए एक्टिव फॉर्म दैट इनिशिएट्स बायोलॉजिकल रिस्पॉन्स। बायोलॉजिकल रिस्पॉन्स को इनिशिएट करता है। व्हेन PFR इज एब्जॉर्ब फार रेड लाइट इट कन्वर्टेड टू द PR फॉर्म। मीन्स ये PFR जब फार रेड लाइट एब्जॉर्ब करता है तो ये दोबारा कन्वर्ट हो जाता है PR फॉर्म में। Degradation of PFR is known to occur by a highly process in which PFR is first attached to small protein called ubiquitin. Attachment of ubiquitin to PFR or to other proteins targets them for degradation and carry out degradations. ATP and 3 enzymes are required. They are: number 1, protease recognize the protein. Number 2, hydrolase targeted the protein. Number 3, release free ubiquitin. Then chemical changes occurs in phytochrome mechanism in plants. Chemically, phytochrome is a homodimer of two identical polypeptides, each with a molecular weight of 120 kDa. Each polypeptide has a prothetic group called chromophore that is attached via a sulfur atom and a cysteine residue of a polypeptide. This chromophore is an open chain tetrapyrrole similar to the photosynthetic phycobin pigment of red algae and cyanobacteria. The chromophore notes the protein that absorbs the light and causes phytochrome responses. When PR is converted to PFR by red light, there is apparently a cysteine isomerization in chromophore. Alternation of chromophore in phytochrome then cause several unidified subtle changes in the structure of phytochrome portion. Then we will discuss about the phytochrome mediate physiological responses. मीन्स mediate जो physiological responses होते हैं वो क्या होते हैं in phytochrome. Phytochrome has been found to control a number of physiological responses of the plant, e.g., spore germination, chloroplast movement, elongation, bud dormancy, root development, etc. Numerous plants' responses are mediated by phytochrome, hence, for convenience of study, they have been divided in positive, negative, and complex responses, or as developmental and rapid responses. नंबर वन, पॉजिटिव रेस्पॉन्सेज। तीन रेस्पॉन्सेज जो हैं यहाँ पे हैं, पॉजिटिव रेस्पॉन्सेज, नेगेटिव रेस्पॉन्सेज एंड कॉम्प्लेक्स रेस्पॉन्सेज। पॉजिटिव रेस्पॉन्सेज characterized by initiating or increase of biosynthetic or growth process, e.g. phytochrome mediate anthocyanin synthesis. Number 2, negative responses, those involving inhibition of growth process or other physiological processes like translocation of substance, e.g. inhibition of hypocotyls, elongation by light. Number 3, complex responses, those which are characterized by initiating or an inhibition and later promotion of the responses as compared to corresponding dark control, e.g. light controlled regulation of oxygen uptake by cotyledon. The role of phytochrome. Many flowering plants use it to regulate the line of flowering base and the length of the day and night photoperiodism and to set circadian rhythms. It also regulates other responses including the germination of seeds, elongation of seeding, the size, shape and number of leaves, the synthesis of chlorophyll and the strengthening of the epicotyle or hypocotyle hook of dicot seedings. Then plant pigment of phytochrome. A plant pigment is any type of colored substance produced by a plant. In general, any chemical compound which absorbs visible radiation between about 380 nanometer violet and 760 nanometer ruby red is considered a pigment. There are many different plant pigments and they are found in different classes of organic compounds. Plant pigments give color to leaves, flowers and fruits and are also important in controlling photosynthesis, growth and development. Phytochrome is a blue-green plant pigment which regulates the plant development including seed germination, stem, leaf expansion, pigment synthesis and flowering. So that is all about the phytochrome mechanism in plants. So thank you everyone for listening my presentation. थैंक यू। अगर आपको मेरा ये लेक्चर आपको समझ में आया है तो लाइक ज़रूर करिए और सब्सक्राइब करके बेल आइकन को ज़रूर दबाइएगा। थैंक यू। प्रदीप सिंह राजपूत।